What we're going to do now is to give you a quick overview of the biology module. And so, as before, we're going to start with products, take a look at the concepts that are involved in the products, and then take a look at the role of the engineer in those products. So let's talk about biology module. As a reminder, in the overview, we could talk about two important products. One is insulin, the other one is penicillin. Okay, so what is insulin? Insulin helps transport glucose into the cell, and type 1 diabetics cannot make insulin on their own. So as to how insulin transports sugar into the cells, we need to understand things like ligand-receptor interactions. So we'll talk a little bit about what they are. Essentially, you said you have a molecule in solution binding, attaching itself to another molecule, and then facilitating the transport of glucose into the cell. So we'll talk a little bit about, therefore, the importance of insulin. Then the question is, what is the role of the engineer? If you need to make insulin for patients, for example, how do you make them? Do you use a chemical process? Do you use biological process? What is the safest way to make them? And then to make them at a price that people can afford. That's also very, very important. And the chemical engineer has played a very, very important role in the production of insulin. There is a difference between the insulin, as we understand, it's a biochemical, and it's different, if you will, from, say, some chemical that we know, like water, alcohol, ethanol, and so forth. But still, it's a chemical, it's a biochemical. So we need to understand the difference. What is the difference between insulin as a biochemical versus other chemicals, for example? The one key difference is, for example, biochemicals tend to be very sensitive to temperature. So the engineer has to understand how to design the temperature so that the system, so that the material, the property of the insulin will be preserved. So as I mentioned in the last slide, we're going to take a look at a little bit about cells and receptors and how they interact with each other and so forth. And then the engineer's role, as I mentioned, is decide on the method of production and how to scale it up. How do you make large quantities of something like insulin, which is thermally sensitive? It's a challenging problem, and we'll talk about some of the concepts that engineers have worked out working with biologists in making this actually possible. Just a slight, this is something we'll cover a little bit more as we go forward. Insulin is a good example of what's called a polymer. Poly means many, mer means one unit. And so, for example, you've heard of polyethylene. Polyethylene is many molecules of ethylene joined together. And insulin typically is essentially polymer. That the mer refers to an amino acid. So there are multiple amino acids joined together. And so insulin is an example of polymer. DNA, for example, is a polymer. It's a polynucleotide. Insulin is a polymer. It's a polyamino acid, and so forth. So the term poly refers to multiple things joined together with essentially chemical bonds. The difference, of course, between polyethylene and polymer is what those bonds are, how they are made, and at what temperature they're made, and it's all the things you need to worry about. So insulin, it's a peptide, and you may be able to see that on the slide very well, much better than on the screen. This is an example of a bovine insulin. There are two chains that are hooked together with the help of these disulfide bonds. And all of these things, don't worry if you don't understand the details right now. These are individual amino acids linked up together, covalently bonded together. And that's the insulin structure. Just a little bit, we won't talk too much right now. This is a good cartoon about how insulin actually works. Insulin binds to something called an insulin receptor on the surface of the cell. That triggers a bunch of events inside the cell and results in ultimately transport of glucose into the cell. Okay, so as I mentioned earlier, if you have to make something by synthesis, the question is, do we use chemical synthesis or do we use biological chemical synthesis, as it were, use some bug to actually make our protein? In some cases, we have a choice between the two. And the one that will win out is the one that's going to be more economical and safe and much easier to use. For insulin, the biological synthesis is going to be much, much cheaper. And that's what I actually use. There may be some links you may be able to follow through diabetes. I'll encourage you to read up about diabetes and the role that insulin has played in terms of helping people with diabetes live a normal life. And it's also fascinating to read about the discovery of insulin. That's something you may actually want to take a look at, as to how actually it took quite a while before people figured out that there is this molecule called insulin that can actually treat diabetics. So I'd encourage you to read up about insulin. And we can have a discussion about this also. Okay, so the biology is that the next product in biology we'll talk about is penicillin. It's a wonder drug of World War II. It's called an antibiotic. It kills bacteria used to fight infections. And there are many different kinds of antibiotics. There are some antibiotics that work by destroying the cell wall. Other antibiotics work by stopping the production of proteins or preventing the DNA from replicating itself. There are many, many different ways in which antibiotics actually work. So we'll take a look at some of those concepts. The problem, of course, is that how do you make these antibiotics in large quantities? As you can see in the structure, the penicillin molecule is very different from the insulin molecule. So the question is it possible to synthesize penicillin by chemical synthesis? And the answer is yes, you can. But it turns out even penicillin is actually made using biological processes, because it is much cheaper and safer to actually make it. So if we actually use a bug, essentially, to make penicillin in large quantities, almost like what Alexander Fleming actually discovered penicillin by growing these little bugs, fungi, mold as it were, in culture plates. Except that the engineer has to figure out how to grow large amounts of these bugs and mold in large quantities and make them and purify them. So that's a substantial challenge too. So penicillin, it kills bacteria that infect and kills humans without harming humans. It's a truly miracle drug. I mean, till a few decades ago, people used to die of ordinary infection. Today, we don't even think about it. It's because we have these wonder drugs, antibiotics. So there are different concepts. We'll talk about antibiotics will work by destroying bacterial cell walls or inhibiting protein synthesis and so forth. And then we will describe how some key developments in chemical engineering enable the production of penicillin in large quantities, submerged fermentation, liquid extraction, all of these amazing unit operations that chemical engineers came up with. So insulin and penicillin are very different chemical structures, very different functions, but and also these are biomolecules, biochemicals. So the reason chemical engineers are involved in this is because really chemical engineers had to figured out ways to make these in large quantities safely and at prices that people can actually afford. So if you go back to the original video, introductory video, we talked about how biology is an enabling science. Today, I'm going to only talk about insulin and penicillin in this course, but if you do some reading on your own, you'll discover that chemical engineers are involved in the production of a number of these biomolecules out there. Molecules like erythropoietin, there are anti-cancer drugs that are made of, that are essentially biological molecules for which chemical engineering production are actually needed. So hopefully you'll get a much better appreciation of why biology is very important in the study of chemical engineering. So that's what we'll talk about.